Hi, folks. It's me again. Hopefully you have already watched the recording for cytoskeleton or cellular processor dynamic. That's part of the pre-work. Um, if you've watched that recording, then you can read chapter 16, these designated pages. And what I would do is just read these pages to reinforce the topics that I covered in the recording. So either I did specific call outs like visit this, figure or this panel, or to reinforce concepts about actin remodeling and microtubule remodeling and ways to regulate their behaviors, for instance, and not try to, read word for word try to memorize every concept. That's not what we're going for this chapter. It's really just to back up some of the sort of main points that I mentioned in the cytoskeletal recording. Okay, as part of the pre-work, I'm making this video, which is based on the content in chapter 9, which is looking at cells and molecules in the light microscope. Last year, I asked the students to just read this chapter, and the feedback that I got was that it was super dense. There's maybe more physics in it than one would want. So what I've decided to do is just summarize the main concepts within this chapter, and then you don't have to read it. However, if you do want to learn more, of course, you're always welcome to supplement this video by reading chapter 9, pages 563 to 588. But what I actually did is I took the figures from that chapter. So Janet Lindsley told me that in this new edition of Molecular Biology of the Cell, so 7th edition, that the authors really tried to use the figures as the narrative. So as you go through the figures, it basically tells you the story and most of the content in each of the chapters. And I will say that they did a really great job with that. So what I decided to do is lift the figures from chapter 9, which is this one, and just tell you sort of main concepts and facts about a particular imaging platform or why you would want to use it. And the goal is for you to understand, if you have a specific biological question in mind, what kind of imaging modality might you use and why? With a better understanding then of when you read papers, you understand why they use that particular platform versus another. Okay, so to begin then, we need to talk about scale. And all of the images, again, that I'm going to show you are from chapter 9 in the 7th edition of Molecular Biology of the Cell across these pages. So when thinking about scale, I think this image shows it beautifully. If you start off with what you can see with your naked eye, so you can see your thumb, you can see the ridges, and then we start to get at a scale where you need assistance. So this is on the tens of millimeter scale. This is, 2 millimeters. And if we go down in order, you can start to see with a microscope the stratification of the individual skin layers. If you go even smaller now to the 2-micron scale, you can begin to visualize and resolve organelles within a particular cell, and then down to protein structure at 20 nanometers to the atomic scale at 0.2 nanometers. And we have different microscopes to be able to visualize each of these different scales. So, 20 millimeters, you don't really need a microscope. Maybe for some of you, you need glasses. And then, when you get to the organelle level of the micron scale or the hundreds of nanometer scale, you can begin to detect these structures and resolve these structures with a light microscope. Smaller than that, when you get to 20 nanometers, and I'll go into why that's the case, we start to require super resolution fluorescence microscopy, and then down to the 0.2 nanometer scale, at the ultrastructural level, we need electron microscopes. And I won't be talking too much about electron microscopy in this short video. I'll mostly be talking about various different kinds of light microscopy. So just by using regular old transmitted light and by bending the sources of light, you can actually detect quite a bit in a cell. So this scale bar is 50 microns across. Here's an individual cell. There's the nucleus. There's the outside of the cell. And because certain structures in a cell will either absorb light or scatter light, based on those changes in wavelengths of light or the phase of light, we can begin to resolve subcellular structures in the cell. So these are actually 4 different kinds of just transmitted light microscopy, either by changing the pattern of the light or by different detection patterns. And we can see actually quite a bit, like the nucleus, and maybe this bumpy stuff looks like it could be the ER. That's for a cell that's pretty flat, right, on glass. What if you have, a big 3-dimensional structure where you want to look with light microscopy? Well, you can section those samples by slicing it over a really sharp blade, and then putting those slices on a cover slip, and then visualizing those thin sections with microscopy. And this has been really great for thick samples or tissue or tumor slices that would be really difficult to resolve otherwise. So here's an example of a section taken from a tissue, and just using dyes that are selectively taken up by various structures and by properties of the different tissues, cell structures, you can resolve actually quite a bit in terms of the nuclei and the outside of the cells, just with light microscopy. This is not fluorescence. These are just different dyes that can be picked up. But because it's so thin, you can resolve like quite a bit in this section. But most of us use fluorescence. So I was talking about transmitted light, which is just regular white light. What about fluorescent proteins? Well, the way that the fluorescent proteins work is that when you excite that fluorescent protein under a certain wavelength of light, you get a photon that absorbs that light, and then it comes crashing down and it emits a photon at a longer wavelength, and that's what's detected. So maybe many of you have heard about excitation and emission spectra. That's shown here. That means for a given fluorescent protein, this looks like it's for GFP, that there's a peak excitation around 488 nanometers. So if you shine 488 nanometers of light, you'll get lots of absorption of the photon. And then during emission, it emits at about 525. But you can see that it's not like a strict cutoff. There's sort of this distribution of emission, this distribution of excitation. So let's say you had 450 nanometers of light that you wanted to excite your GFP with. It would still kind of work. It just wouldn't work as great since it peaks at 488. But the idea is that these fluorescent proteins are great because you can hit your sample with different lasers that comprise of different wavelengths of light, and then you can detect different fluorescent proteins in your sample sequentially. So here's an example of, this is not actually GFP or RFP. These are different dyes that are selectively taken up by structures in the cells that are conjugated with a color. And it's an example of using different excitation wavelengths. You can detect, what's the green structures, and using a different excitation wavelength, you can detect what are the red structures. We have lots of different fluorescent proteins that sort of span a lot of a range of wavelengths of light, starting from DAPI, that's sort of on the shorter wavelength, to Psi5, which is on the longer wavelengths of light, and a lot in between. So that's with dyes that are taken up by cells. What if you wanted to look at a specific protein as an example? And you knew that there was an antibody that was generated against that protein such that you could visualize it. So that's shown here by this little red triangle, which is, antigen A. You can use that antibody that recognizes antigen A, and it will specifically bind to that antigen. And then you can take a secondary antibody that's coupled to something that you can detect, like a marker. Sometimes it's a dye, sometimes a fluorescent protein. That secondary antibody will then detect the primary antibody. You can see kind of this amplification of signal too, to then look at localization of this antigen, this red triangle, in a specific cell or in a specific tissue. Antibodies are quite a bit bigger than dyes, so they're not really great at getting into cells when the cells are living. However, they can detect antigens that are on the outside of cells. So if there's like a receptor that you're interested in and you have an antibody against that receptor, those cells could be live and you could detect the presence of that receptor on the surface. But if you wanted to detect an antigen that's inside of the cell, typically those cells have to be fixed. So you can, they're dead, and you could poke holes in them such that the antibody can get into the cell, just simply because they're bigger. So if you combine these two ideas, taking a tissue and sectioning them, and then using specific antibodies to detect antigens of interest, this could be a really powerful technique to say, visualize a protein of interest within a particular tissue. But that's for, really thin tissues. What if the tissue or the specimen is fat or thick? So this is a C. elegans embryo. It looks like it's kind of flat, but it's actually this three-dimensional, embryo. So it's round. It's like a football shape. You can see with conventional microscopy, it's just too thick. I mean, you're fluorescing, in this case, GFP molecules inside the embryo, but also on the outside of the embryo. You're getting lots of out of focus light because it's just a fat tissue that you're trying to image through. What you can use instead is a technique called confocal microscopy that allows you to remove all of that out of focus light. And then you can resolve an image like this, which is taking a single slice through that embryo. So how does that work? Okay, so if your sample, here's your sample, the red thing, it's pretty fat, and you want to actually focus on something that's right in the middle of that sample, what you're going to get is, you're going to try to illuminate that part and detect, the emission of the photons, but you're also going to get everything around it. What you need is a confocal such that all of your excitation basically happens at a single point of focus. And it's usually on a plane that's planar to your sample. So in the z-axis, the axial direction of the objective. When those photons get emitted, your detector goes through a pinhole, which also helps detect only the focus light and not any of the out of focus light. So what this example is showing you is you just had a point of excitation right here in the middle. So all the stuff that gets emitted then gets picked up by the detector. All the stuff that wasn't supposed to be collected, like up at the top here, is now missing, right? It's like on the outside of what's being detected. So what these pinholes, these confocal pinholes allow you to do is focus in the laser so you can really excite a specific point of focus, as well as detect a specific point of focus. And as a result, remove all of the out of focus light so you can get better resolution. So if you did that through the sample in that axial direction, in that Z direction, you could begin to then reconstruct it back into 3D with software. So that's shown here, a network that, in any individual plane, you would just be taking a slice through it. But as you go through that specimen with confocal imaging and reconstruct it, you can reconstruct the 3-dimensional image. Okay, what about samples where, it's not that fat of a specimen? It's just an individual cell that's shown here, this cell, but you really want to illuminate a plane that's pretty close to the glass. So really close to the bottom of that cell. And you're not really interested in all the fluorescence that's inside of the cell. So this sample, again, is not very thick. It's just one cell. So this whole peach thing is supposed to be one cell. But you're really only interested in the molecules that are 200 nanometers from the cover slip of that cell. Well, there's a certain kind of platform for that. It's called turf microscopy. In general, what it does is it changes the angle of light for that excitation emission wavelength such that it only illuminates 200 nanometers from the cover glass, and that's also what it detects. So that if there are these structures that are really small but close to the glass that you wanted to resolve, such as here, you can do that and get good signal to noise because you're not going to pick up anything else that's deeper into that cell. And actually, we're going to talk about this in class in an example of where turf microscopy is really handy. So all of this talk about resolution, what actually determines resolution? What determines the resolution is the point spread function. And what this means is if you illuminate a sample, its highest point of illumination is in the middle, and then it sort of drops off on each side as a Gaussian distribution. And that's in all directions, right? So it's highest in the middle, and then it slowly drops off toward the edges. It means that if you had 2 points that are really close together, because of that point spread function, they would begin to overlap. The limit of resolution for most light microscopy is 200 nanometers. Because if you start to, and that means like if there's a sample here, like a GFP molecule here and a GFP molecule here, you might be able to resolve them as 2 separate GFP molecules if they're 200 nanometers apart. As this circle gets closer and closer to this circle, it's going to look like one circle, right? Because the point spread functions are overlapping. And it's actually that determines what is your resolution limit. If you're looking at 2 things that are less than 200 nanometers away, it's going to be very difficult to resolve those them as 2 different structures with just regular light microscopy. Luckily for us, there are strategies in place that are called super resolution microscopy, which gets beyond the challenges brought up by the point spread function. I'm just going to talk about a couple of examples of here. There are obviously more. One is called STED. The idea behind it is you have an excitation spot, but then you have this like donut of depletion, such that whatever is left behind is right in the middle, and that's your effective fluorescence. So your point spread function essentially gets a lot narrower, and you could begin to resolve structures that are closer together. The other idea is if you have this like plane of illumination, as in, so here, these are filaments, right, that are, let's say they're all GFP expressing. And I wanted to know the individual like monomers in that filament. That'd be pretty hard to see if I just lit up everything like this, right? Like everything is green. But if I gave it a very low level of laser light, such that stochastically, randomly, GFP molecules would excite, but it would be random. So let's say in this interesting shape structure here, you know, when I use low level of light, and only some of the GFP molecules excite at one time, and then at a second time, these GFP molecules excite, and the third time, I can begin to reconstruct the whole thing because I illuminated only a little bit at a time. And now what you can see is sort of the individual structures that make up this string versus if I just illuminated them all at the same time. That's called palm and storm. And it's another form of microscopy that gets around the point spread function because we're not going to illuminate its GFP friend that's right next to it. We're going to illuminate a GFP friend that's maybe a little bit further away. So this successive cycles of activation and bleaching then allows you to reconstruct what is ultimately the final expression pattern of that fluorescent molecule. So again, the point is to say that you can resolve structures that are closer together than 200 nanometers. It just requires super resolution techniques. So that is sort of a synopsis of the kinds of imaging platforms that you can use to help you image across scales. And I'm going to end with just a couple of example questions. So if you wanted to look at the localization of protein X, let's say in a tumor, what kind of sample processing and imaging might you consider? There's no one right answer because there are so many different platforms, and it really depends on sort of what is your goal for this experiment. If it's simply to ask, where is it? You might say, okay, what I'll do is first I'm going to section the tumor because I want them to be nice and thin. And if I have an antibody against my protein X of interest, I'm going to use my antibody to detect where it is in that tumor section, and then I'll know where it's localized. And because the tumor sections are pretty thin, I can basically use any fluorescent microscope because I'm not worried about the thickness of the sample. So that's one way. Maybe another way is I want to fluorescently tag that protein of interest. So I make protein X GFP, make the tumor, isolate the tumor. Now the tumor is a little bit thicker. So I'm going to use confocal microscopy to visualize the GFP localization in that sample because my tumor slice is a little bit thicker. Maybe I could even keep part of the tumor alive and use live microscopy because I GFP tagged it, right? That tells me the localization, but also tells me if that localization changes. Or maybe you'll use that same fluorescently tagged protein of interest, right? Protein X GFP, and then cut sections instead. So now it's like fixed. I'm cutting sections, it's thin. I could look at really high resolution where that GFP type protein. My point is there's lots of different ways to do this. It just depends on what you want to get out of it. Okay. Here I have also, because you can use an antibody. Okay, what if you had some evidence to suggest that that protein X actually localizes to the inner mitochondrial membrane, and you wanted to ask if that's true in a tumor? Okay, the inner mitochondrial membrane. Remember, when you look across scales, this little nugget mitochondrion is about 2 microns. And if you want to look at the inner mitochondrial membrane, you're probably in sort of the 200 nanometer, maybe less range, because you need to be able to resolve the difference between inner and outer mitochondria. So what kind of imaging platform would you use for that? Bearing in mind, what is your limit of resolution for a light microscope based on the point spread function? So what kind of imaging platform might you use? So think about this. We could discuss it further in class. Ask me any questions that you have. But the idea is for you, again, to be thinking about, can you be familiar enough with microscopy techniques to really understand why a particular technique was used in a paper, or what kind of technique you would use in your research? Okay, so again, this is the recording for chapter 9. If you want more information, getting more into optics, feel free to actually read the chapter. And then don't forget to also watch the cytoskeletal recording and do the associated reading. Okay, see you in class.